early 90s, and there were no genome sequences at the time. So we depended upon information that was flowing out of the MRC laboratory in England, where John Sulston and another Nobel Prize winner and Alan Coulson were assembling the kind of map we needed to find these genes. And so there was a wonderful communication between the MRC laboratory and our labs, my lab, Gary Ruvkun's lab, and others, where we were daily receiving information that was bringing to get into sharper focus the map of the genome, the DNA of C. elegans. And that greatly facilitated our finding the Lin4 gene. And we knew we had the Lin4 gene because that mutation that we were talking about broke the sequences of the DNA where there was a small RNA being produced. And that small RNA has been come to be called the Lin4 RNA. It was the first microRNA to be identified. It's called a microRNA because it's really unexpectedly short. It was surprising that there could be enough information contained in only 22 bases or code words in the genome for this microRNA to regulate another gene so precisely. And it turns out that we now understand how that works because the microRNAs are part of a broader suite of regulatory mechanisms that were the subject of the studies that Andy Fire and Craig Mello won their Nobel Prize for, which is RNAi, RNA interference. And the important point there is there's an ancient protein called an argonaut who's evolved to latch onto these tiny sequences of nucleic acid from these genes called microRNA genes. And that flies around in the cell and finds matching sequences in the products of other genes. So what the microRNAs really end up revealing for us is a way that parts of our genome can communicate with other parts of the genome and regulate the activity of those other parts in a coherent way that enables our cells to manage the processes of development, tissue renewal, response to physiological changes, and so on. Another layer of regulation of the gene regulatory apparatus. It's fundamental to all of us, animals, plants, and other living organisms. The microRNAs are found in plants and animals. And what else can I say? Is that good?
Like I understand
Yeah.
science, but we have a question.
Or was that comprehensible?
Perfect.
Yeah. Thank you. Oh, I was astonished and surprised, delighted. Everything you might expect. I honestly, this was not something that I expected, to be quite honest. And so, because in my opinion, the Nobel Prize to Mello and Fire encompassed all these phenomena that we studied. So that was my my sense, and I was comfortable with that. So as you can imagine, I was astonished by this. Yeah. The question was, no, can you repeat the question? Thank you.
Thanks for giving me a purpose. The question was for people around the world who are for the first time today hearing about mRNA, what is it that you would like them to know about the science or about its potential?
Yeah, I think one of the messages, honestly, that I would hope to convey to anybody who's listening is that these kinds of studies of laboratory organisms, like in this case a nematode, Caenorhabditis elegans, are critical and key and fundamental to advancing understanding of biology. And I mean advancing understanding of biology in a way where we are constantly moving into territory that was unexpected. And I think the unexpectedness of biology is probably the most important principle, perhaps, for people to appreciate. Living things are the way they are because they evolved this way. And the processes that bring living things to the state where as we see them today, are poorly understood. And the underlying processes in our cells and the processes that manage and coordinate the action of an organism like this are still deeply mysterious. We know a lot, but in a way that can seem like an illusion, that we know so much. We all have the genome sequences, the DNA sequences, and we identified thousands and thousands of gene products, whether they're RNA gene products, protein gene products. And so at any given moment, it feels like we know most of what we need to know, but that is actually an illusion that we have to consciously disabuse of ourselves of and leave ourselves open for the surprises.
Any other questions?
Please describe the significance of your discovery to, say, a farmer in Ohio.
Farmer?
To a layman, to a regular Joe.
Yeah, I think the significance of this discovery of microRNAs is that it allowed us to be aware of a very complex and nuanced layer of regulation whereby genes in our cells talk to each other and coordinate their activity, and in ways that enable those cells to communicate with each other, to send and receive signals throughout the body. This is a mode of regulation of gene activity that adds to all the other layers of gene regulatory activity on the level of production of RNA products, on the level of turnover of gene products, the modifications of gene products that happen by other gene products. So the products of our genes are talking to each other in an incredibly diverse way of mechanisms, right? And the microRNAs is one of those layers, let's say, of communication amongst genes. So the importance of finding it it goes along with the importance of finding any of those other layers of gene regulation, because they all matter in different contexts, in different ways.
Yeah. The question is, on a personal level, how significant is this Nobel Prize recognition to you?
Well, yeah, I really haven't collected all my thoughts on this question. In the moment, I'm very aware that this presents an opportunity for me to speak to some of these issues that I've been trying to address, or you mentioned about the importance of public support of research, the importance of the best kinds of leadership in institutions, the importance of collegiality and the sharing of information amongst scientists. All these principles that I find so important for the advancement of science and that were so important for my success, my career. I want to shout out to the world that this is really important. All these things are important. So, the other thing is, it gives me an opportunity to mention that I benefit from the people who came before me. I mentioned my parents, my father, mentors along the way, you know, Ed Gruberg, my postdoc who mentored me when I was an undergraduate and taught me how to, if you don't know how to do something, walk out the door, down the hall, see if you can find somebody who knows how to do it, you know? Before you write a paper, go read all the literature, something I probably wouldn't have known about. These fundamentals, so you look back and see the folks who taught you the fundamentals. David Baltimore, my PhD advisor, who showed me that, a simple experiment, carefully designed, strategically connected to an interesting and important question can yield a lot, David also taught me indirectly that you can have all kinds of different attributes as a human being and all kinds of shortcomings, as a scientist, and it's still okay. You can contribute, right? Because David Baltimore is a man who's absolutely brilliant and inspirational, and I could look to him as somebody who I knew and knew that I was not brilliant, but I could do good work, and make a contribution. So I think that one of the important messages it occurs to me to say is that, often we are standing around amongst really amazing people. If we're scientists in an institution like this, everybody around us seems really amazing, and often you wonder, what am I doing here? And and so I've come to realize that I'm here because there's a few things I can do okay. There's a zillion things I can't do okay, but I work in this, things get done. I can work with folks who have complementary abilities, and talents and stuff like that. I don't have to be the smartest person in the room. I just have to be in the right room, and I think my career has been characterized by luck of being in the right room,
Victor, how did you learn the news this morning?
Am I what?
How did you learn the news?
Oh, yeah. So this morning I was sleeping late, and my phone was downstairs, and we get a call from my son, who calls my wife, Candy Lee, and says, if you get a call from somebody in Sweden, answer it. Because they had called him. So I guess the poor people over there were getting desperate, and they started, let's say, all right, anybody Ambros in the general area.
And what's it like getting that call? What do they say to you?
He said just that. Answer your phone. What did the Nobel people say? Oh, I mean, they were very nice. I mean, they said that they were really happy about this, recognizing this discovery, and they were all excited. So, I took that to heart. I think they were, yeah.
Thank you. Any other reporter questions? The question is, after a few hours of the news sinking in, what's going through your mind now?
Well, quite frankly, I'm enjoying this moment right now. Being here with you, and I'm looking forward to later in the day getting together with Gary Ruvkun, if possible, because that'll be a fun celebration. That's what's going on right now. Yeah. Thank you.
Anybody else? Henry.
I have several questions, but There were several questions about from a lay perspective. What potential applications do you see this potentially leading toward in terms of therapeutics? Also, I find your shirt very interesting. You could have worn anything this morning, and you elected to wear that shirt with tigers.
Yeah.
So do tigers mean anything to you in terms of this special moment?
Okay, I can answer the second question first. This was a shirt that my wife, Rosalind Lee, bought for me a couple of months ago in a secondhand store. And it's become my favorite shirt because I get comments about it. It's the only, honest to God, I get the average of 2 comments a day from strangers. So, yeah, so it has wonderful provenance because Candy bought it, and I like to brag that it was bought at full price, which is about $5, right? Yeah. So, yeah.
And the first part of the
First
question
is significance of the finding of the discovery of microRNAs with regard to improving human health. Well, I mean, the most straightforward thing to say is the deeper we understand living systems, the better equipped we are to figure out what's wrong when these systems go awry in the context of disease. Yes. So thank you. So lately, I was quite astonished actually to realize an aspect of this issue of relevance when we attended a meeting in Copenhagen of the, it's an Argonaut meeting. So it's all about these Argonaut proteins that Craig discovered were the key partners of small RNAs, including microRNAs. And at that meeting, there was a number of cutting edge talks that were advancing in amazing ways and astonishing ways our understanding of how this wonderful protein works, But these studies, the most amazing studies, were from people analyzing mutant proteins where the mutation in that Argonaut was identified in a family where the child has what's now called Argonaut syndrome. So these are families where the individual, the youngster, shows developmental delays of various kinds. Sometimes, complex developmental delays. It can vary a lot from patient to patient, and in every case, that patient, the child, has a spontaneous mutation in the gene for one of their Argonauts, the microRNA Argonauts. And that mutation was not inherited from the mother or the father. It's a spontaneous mutation. And it turns out these mutations are in amino acids of Argonaut that are conserved in C. elegans. So our lab and these other labs had started to take those same mutations that were found in the human families and put them into the Argonaut of C. elegans and try to learn what we can about how this protein is broken, And as I say, that several labs reported really amazing findings that, on the one hand, were telling us giving us new insight into how this amazing protein Argonaut works. So the basic sciences in the audience were learning from the mutations that were presented to us by the families. And several families were there at the meeting, and several of the children were there, and we got to meet them. And for me, this was an amazing experience because I was, for the first time, trying to process this idea that we basic scientists are learning from these patients, right? And so, and then there was a really interesting discussions at the meeting, of course, about how we basic scientists could end up somehow helping these families, if not now, in the future. And so there was talk about potential therapeutics, but the discussions were actually collaborative discussions between the patients, families, and the scientists in the room, you know. So it was I think that, I guess it's a long-winded way of saying that the significance of this kind of work to human health, to me, is probably, it's unexpected. Of course, there's companies out there, obviously, who are using small RNAs as therapeutics, and there's a big thrust here, and some amongst those small RNAs are microRNAs. So it's a very active area of therapeutic development in industry and in academia. But I wanted to tell you that story because it was so personal and so interesting for me. And it actually, then you start to think about how can I shape my research in a way that I can continue to, uncover fundamental things if I can, but also to try to think and along this other angle at the same time.
More questions? If not, why don't we get our two Nobel
Oscar
laureates?
has a question.
Oh, I'm sorry. Okay.
Oh, what to say to the science trainees or younger generation? So those, if any of you are in the room who are science trainees or younger generation, you know that I can really become a flamer sometime and really, blather on and on about, I mean, the basics, the basic thrust of it is, you guys, each one of you is talented. Each one of you has something to offer, not just something really special and important to offer, and the field that we study, let's say RNA biology, if we could put it that way, is a field that's just incredibly exciting right now and will continue to be, indefinitely. So keep up the good work and just know that you are the right person for the job you're doing, and you're going to do great things.
Thank you. If you could please thank your son for calling you. No one's happier than we are that you're here. So let's get our two Nobel laureates together. Craig Mello, co-recipient of the 2006 Nobel Prize.
Sure. Oh, I mean, what a long overdue and wonderful day it is. They found a way to surprise both of us because I was surprised that they had awarded the prize to us, skipping over Gary and Victor for now almost 18 years or so. So it's kind of a relief, to finally feel like justice has been done. The discovery that Victor and Gary made was an inspiration to me. Also, of course, Victor was my mentor and Gary was on my thesis committee. So I knew them both very, very well, and they both inspired me for different reasons, just full of enthusiasm and curiosity, really believing in this little organism and what it had to teach us, and being right there with advice along the way. Victor's just a tremendous mentor, and I had the pleasure of finishing my PhD in his lab. And without his help, I don't think I wouldn't be here without Victor. Victor really made it happen, believed in me enough to help me get through a really difficult time in my own career when we were trying really hard to get, micro injection of genetic information back into the animal to work. And it didn't work. But with Victor's help, we really got it working. And that's sort of, so I feel really emotional right now to be here to celebrate Victor. Just an incredible, inspiring person. He's the kind of person who always has an insightful question at every single meeting that you go to. He sparks all kinds of ideas constantly. I also want to mention that Victor was a co-founder of the UMass RNA Therapeutics Institute, which really, again, we owe great thanks to both the Nobel Committee and to the state of Massachusetts for investing in our institution. After the 2006 Nobel Prize, the state came up with $100 million that helped make this building possible. And in so doing, also created the foundation for an RNA Therapeutics Institute, which would build on the expertise in RNA biology that Alan Jacobson and others, Melissa Moore, Phil Zamore, Michael Green, had established here at this institution. And so it was extremely timely that that institute was named the RNA Therapeutics Institute. At the time, there were no RNA therapeutics, but it was a clear aspiration. And again, Victor is really too humble he should take credit for the therapeutics because they use his mechanism. The therapeutics actually that are made today are short pieces of genetic information, just like the one he discovered first, Lin4. And we can now make these, we call them search queries because they program a search engine, which is this Argonaut protein. It's the Google of the cell. And for those in the lay audience out there, imagine these things can search inside of our cell and find matching information. And they do it at 100 million collisions per second. They're incredibly efficient. They're really fast. Our body is doing computations all the time. And we know like really this much about how organisms handle information. And yet it is fundamentally important to human health, human biology, and the future agriculture. I mean, there's so many applications for improved understanding of how our body handles information. And everybody knows about DNA, and I guess the whole world got a crash course in RNA. Now, sometimes Victor will get this too. I've been given credit for discovering mRNA. you will get it. mRNA is different, okay? These little RNAs in a way have much better therapeutic potential because they're tiny, they're easy to make. They don't need to be put into a compartment and delivered inside of a shell. They can be delivered directly. And one of the great things that Victor and I and others here have been able to do is recruit some really amazing RNA chemists who have been helping us and the community make these into drugs that are more effective and deliver now really throughout the body. So you guys, the lay people out there, more and more of you are going to be getting these kinds of medicines, and we can only hope that they will not only help people with Argonaut syndrome, which I think they can actually, and many other diseases, even cancer, Alzheimer's, pretty much you name it, there's a genetic basis for every disease. And so thanks to the work of Victor and Gary, all of this is now possible. I mean, really, they've just been, you know, at the forefront of a really, really exciting revolution in biology. It's one where, you know, pretty much, as Victor said, we just keep realizing how, with every new discovery, we just keep realizing how little we really understand the nature of how we work and how much more work there is to do. So I'll stop there, and I just encourage any young people, if you're inspired by the work of Victor and Gary, get involved in biology. Biology is having an amazing revolution, and so is medicine. They really go hand in hand now. So anyway, one more round of applause for Victor and Candy.